Hello everyone. Today my poster presentation title is an updated review on the microsponges. First there is the abstract of my poster. Second is the introduction. In this era, medical field has the various advancements for the effective and safe treatment along with the patient compliance. Microsponges are one of the novel approach for the sustained drug delivery. These are highly cross-linked, porous, and polymeric microspheres entrapping broad range of active pharmaceutical ingredients and formulations like powders, lotions, and creams. Microsponges was discovered by Richard Bond in 1987, and the original patents were designed to the advanced polymeric system. And the next is the silent features of the microsponges delivery system. They are stable over the pH range about 1 to 11, thermally stable up to 130 degrees Celsius. They are self-sterilizing in nature, so in formulation there is no need to add the preservatives. They have high payload capacity. They are cost effective as compared to the other drug delivery system. The next image depicts the structure of microsponges. Next is the mechanism. The mechanism is divided into four parts. First is the temperature trigger release mechanism. Due to temperature change, API release throughout this system. Pressure trigger release, when pressure is applied on dosage form or rubbed onto skin and trapped drug get released throughout the microsponges. Solubility trigger mechanism, porous system, which are incorporated into the water-soluble excipients, releases a drug in the presence of water or by diffusion mechanism. pH triggered release, in this mechanism, change in pH triggered the drug release and is achieved by modification of coating on various microsponges. Next are the benefits. There are various benefits, like they improve the efficacy of the dosage form and provides the extended release and improve stability of dosage form. They are non-mutagenic, non-allergenic, and non-toxic in nature. They improve the flexibility as well as the BA of the dosage form. Next is the drug loading in microsponges. The main methods of drug loading are first is the liquid-liquid suspension polymerization, which is also known as bottom-up approach. Next is the quasi-emulsion solvent diffusion method. The image depicts the quasi-emulsion solvent diffusion method. In this method, two phases are prepared. First one is the inner phase, in which drug and polymer solution are prepared. Outer phase is the PVA, polyvinyl alcohol plus water. Both the phases are get to mix. Then the quasi-emulsion droplets formation take place. And by diffusion method, the organic solvent out of droplet take place. In the next step, solid microsponges are formed. Then by filtration and drying process in oven at 40 degrees Celsius for 12 hours, the drug-loaded microsponges are obtained. Next is the characterization. There are various characterization parameters like particle size determination and pore size determination, surface topography, entrapment efficiency, release studies, compatibility studies, resiliency, which is also known as viscoelastic properties of microsponges. Next is the determination of true density. Next are the applications. They have various applications like topical delivery. Microsponges reduce the irritation of effective drug without reducing their efficacy. Furthermore, they are the porous microspheres, so API can be incorporated into various formulations. As an oral drug delivery in this microsponge system, increase the rate of solubilization of poorly soluble drugs by entrapping them in the microsponge system pores. Microsponges technology is also used in bone and tissue engineering, also used in cardiovascular engineering, and they are also used in the reconstruction of vascular wall using the microsponges technology. Next is the conclusion. The novel microsponges drug delivery system offers various advantages over conventional formulation, and has a number of applications in topically, oral, as well as thalamic delivery of drugs. So it serves as an unique and effective technology for controlled as well as sustained drug delivery. Lastly, there is references. Thank you.